I'm Dr. T, and welcome to my office. Let's talk nucleic acids. Nucleic acids are going to be a biopolymer, and they're going to consist of a monomer known as a nucleotide. Nucleotides have three components: a ribose backbone or a ribose sugar in the center. Attached to this is one or more phosphate groups, and on the other end is a nitrogenous base. This is an aromatic base that is then able to interact with other aromatic bases similar to it. The phosphates and the ribose sugars are then attached to each other in order to form the polymer form. In cases where there is a double helix, like that of DNA, then the nitrogenous bases are able to interact with each other with what is known as hydrogen bonding, or that is the attraction of hydrogen that is attached to an oxygen, nitrogen, or fluorine, with an oxygen, nitrogen, or fluorine on the other molecule. This then allows for a rather interesting approach, as that the alignment for hydrogen bonding only works in certain pairs. Specifically, adenosine only lines up with thymine or uracil, and guanine only lines up with cytosine. You can get the other ones to mismatch, but not so easily, and cells do a fairly good job of making sure that doesn't happen. There are two major types of nucleic acids. The first is DNA, and this is actually the least common of them, but one of the first understood because it's somewhat more stable. Specifically, it is missing one oxygen on the ribose sugar, hence the deoxy in its name, and it's used almost exclusively for data storage within the cell. It's essentially the archival grade material that the cell uses to store data. The majority of nucleic acids in the cell are known as RNA, or ribonucleic acid, with the oxygen in this particular case, and these can form a variety of functions: data transmission, enzymatic activity, although we call that a ribozyme since it's not an enzyme because it's not a protein, as well as facilitating the production of proteins, utilizing things like rRNA or transfer RNA, which moves the amino acids around. Additionally, nucleotides are useful for within the cell. Adenosine triphosphate is very common, as it is used as a short-term energy store. Energy is extracted from energy sources such as lipids or carbohydrates, and that energy is used to produce adenosine triphosphate, which is then utilized by enzymes as an immediate energy source. I usually like to think of it in the terms of an energy portfolio. Lipids being long-term energy storage, carbohydrates being medium and short-term energy storage, and nucleic acids, or I should say nucleotides, specifically adenosine triphosphate, but not exclusively, are immediate use energy storage. They're essentially the cash in your wallet. You're gonna go spend it, even if you don't want to, because it's burning a hole in your pocket. With that said, that's a brief and very short introduction to nucleic acids. Thank you, and have a wonderful day.